All right, everybody, welcome to week 3 of online delivery formatting due to COVID-19 shutdowns. And in today's lecture, I'm going to break it up into 2 parts. So this first part is going to be all about nervous tissue anatomy. And then the second part, which will be a separate video, will be all about nervous tissue physiology. So with that, let's go ahead and get started. So again, this part 1 is going to be the anatomy. So we're going to talk about the different divisions of the nervous system, functions of the nervous system in general. We're going to talk about neurons and their structure and how we classify them. And then we're going to talk about neuroglial cells of the central and peripheral nervous system. So beginning our discussion on the nervous system, the nervous system is the master controlling and communication system in the body. And we organize the nervous system into the central nervous system, which is going to be consisting of the brain and our spinal cord. And then the peripheral nervous system is going to be things like our cranial, spinal nerves, ganglia, basically anything that's not brain and spinal cord. So let's talk a little bit more about these things. Central nervous system, as I just said, includes the brain and the spinal cord and is located in the dorsal body cavity. So if you remember way back to chapter 1, we started talking about general terms of anatomy and body cavities. And if you remember the dorsal body cavity, dorsal, like the dorsal fin on a dolphin, it's the posterior body cavity. The central nervous system is responsible for integrating and sending out commands. So the central nervous system is going to interpret any kind of sensory information that it receives. It's going to integrate that sensory information and make a decision as to what to do about that sensory information and send that out via these motor output neurons. So kind of just a quick little picture I just put in here. You'll see this picture again, but interpret sensory input. For example, if you see your glass of water, your eyes are seeing, so that's the sensory information, and your eyes, that sensory information that you're seeing is going to be sent to your brain. Your brain or your spinal cord, either one, the spinal cord often will send the signals up to the brain. The brain will decide what to do about you seeing that glass of water. And say you see that glass of water and your brain decides that you should pick up that glass and take a drink. The brain will send a signal out through this motor pathway to a muscle, causing the muscle to contract, thus you being able to lift the glass of water and take a drink. Okay, so the brain really is the central command of the entire operation of the nervous system as a whole. The peripheral nervous system include things like the cranial nerves, which carry impulses to the brain and from the brain. The spinal nerves, which carry impulses to the spinal cord and away from the spinal cord. And then also the ganglia. And the ganglia are collections of cell bodies of neurons. And so the peripheral nervous system then is basically every other thing except brain and spinal cord, okay? All other nerve-like features minus brain and spinal cord. And the whole purpose of having the peripheral nervous system is the peripheral nervous system is kind of like the communication liaison between the body and the central nervous system. Okay. So it's like the little messengers. It's like the mail workers that take the packages and they come from a central processing center and they get delivered to your house. So the delivery man are the peripheral nervous system. Okay. kind of going along with the peripheral nervous system, it's actually divided into two functional branches. And those functional branches are things that we've already slightly touched on. We have the sensory and the motor division. The sensory division is also called the afferent and motor efferent. So the sensory division is the division that's receiving information from either somatic input, so from skeletal muscle, or from visceral input, which would be from organs. So this is an example of the sensory division receiving information from a visceral input. So this is from the eye, and sending that information to the brain for it to be integrated. Okay, so afferent is moving to the central nervous system. The efferent division is going to take that information that it receives in the central nervous system and is basically just the messenger. So it's going to take that information from the brain. The brain's deciding what to do with the information, and the brain sends a signal out via these motor outputs, these motor signals, which the efferent division delivers that signal to effectors. And those effectors might be muscles, but they might also be glands. The motor division, so this motor output, is actually going to further subdivide into either the somatic nervous system, which is the motor division that controls our voluntary movement, like lifting a glass of water to take a drink. That's voluntary. But then it also subdivides into this autonomic nervous system. And when you see the word auto, you think like automatic. It happens whether we want it to or not. This is our involuntary response. And this autonomic nervous system then has two divisions, the sympathetic and parasympathetic division. So I know this is a lot of subdividing, so I have a graph in here to hopefully make this easier. So we have the central nervous system is the brain and the spinal cord, and it is the integrative control center. And then we have the peripheral nervous system, which are the peripheral nerves, specifically the cranial and the spinal nerves. And the peripheral nervous system, again, is responsible for the communication between the body and the central nervous system. The peripheral nervous system breaks down into a sensory and afferent division, which is going to be composed of sensory information that takes signals from receptors, such as the eye, and delivers them to the central nervous system. And then the motor division is going to be composed of motor neurons, which takes signals from the brain and the spinal cord and delivers them to effectors. And the effectors are going to be either muscles or glands. And this motor division, as I stated on the last slide, is going to then further subdivide into being either somatic motor, which would be our voluntary movement, like picking up that glass of water that you want to drink. And then also the other division is the autonomic nervous system, which is our involuntary responses. And our autonomic nervous system then has two subdivisions, the sympathetic division, which is going to mobilize our body system. So this is our fight or flight response. And then the other division is the parasympathetic division. And this is the division that is active when you guys are just sitting and relaxing and maybe studying. So this is our rest and digest operating system. And I just want to point out that today you're getting a very broad overview of all things nervous system. In the next module, we will focus on the central nervous system. And in 2 weeks, we'll focus on peripheral nervous system. And then the week before the final, we will focus on autonomic nervous system. So you'll get a lot more of each of these bits as we finish up the semester. Okay, so this is just a very general, broad overview. So I've already kind of mentioned these, but I just wanted to again point out this is just kind of a summary of the functions of the nervous system. It has three overlapping functions. We have sensory input, which is the information that's going to be gathered by those sensory receptors about the internal environment and our external environment. Okay, so it doesn't have to be something that you see in the outside world. Maybe you have, I don't know, a fluctuation in temperature. We have sensors that will take that information and send it to the brain and basically tell the brain, hey, brain, I think that there's a slight fluctuation in temperature here. And then the brain will take that information and do something about it and send out its motor signal. Okay, so just so you know, the sensory input, it doesn't have to be an external environment stimulus. It can also be an internal environment. The integration center then is the processes where processing and interpretation of all the sensory input comes into play. And then the motor function of the nervous system is to activate effector organs like muscles and glands to produce some sort of response to our sensory input. Okay. So first clicker question, and as usual, as a reminder, make sure you pause the video to give yourself enough time to answer the question. Don't just rely on a couple seconds passing by and then me automatically giving you the answer, okay? Pause it and really quiz yourself. So on this one, I just want to point out that um, this is an automatic response, okay? Specifically, an increase in heart rate would be sympathetic division, okay? All right, so now that I've just introduced the general concept of the nervous system, we're going to start talking about the cells that work within this nervous system. We kind of are going to start at this microscopic level of talking about neurons and the cells that support neurons. And then again, next week, we'll talk about bigger nervous system structures like the brain, the spinal cord. And then the week after, we'll talk about nerves as a whole. Um, so I just want you to know we're starting small and we're building our way up because the nervous system is so complex. There's so much to know. We've subdivided this into several different sections. So we have a couple of weeks to actually get this information down. So neurons then are nerve cells, and these are the structural units of the nervous system. They are very specialized cells that are excitable, and that word should be familiar because last week we talked about, or 2 weeks ago, we talked about muscle tissue. And muscle tissue is excitable, right? It responded to stimuli. And muscle tissue also transmitted electrical signals. So hopefully you can sort of start to correlate nervous tissue with what you already know about muscle tissue, and that will make your lives a lot easier when learning about this stuff. So neurons are excitable, so they respond to stimuli, and they also transmit electrical signals. Neurons are really special because they are extremely long-lived cells, lasting essentially a person's entire lifetime. They are unique in that they are amitotic, which means they generally don't undergo cell division. So it's kind of what you have is what you have sort of situation. They also operate on a very high metabolic rate. And high metabolism means that they burn energy like crazy, right? And so because of this, they require continuous supplies of oxygen and glucose in order to make ATP. we're going to talk about now the structure of neurons themselves. So neurons have cell bodies, and then they also have processes. And those processes, there's two different types. We have dendrites and axons. So let's go ahead and get into this discussion. The neuron cell body is also called the perikaryon, also sometimes called the soma. But as a whole, the cell body is the biosynthetic center of the neuron. And this is just like think about a regular, just a normal cell. Okay, if we just draw a normal cell, you guys know when you guys draw normal cells, you always put the nucleus kind of, for some reason, everyone in our class always did this whenever I had you guys drawing cells. Um, Remember we had the nucleus, and then you guys like drew in your different structures within cells, like your little mitochondria and so on and so forth. Okay, all of that stuff is cell body. Okay, it's synonymous here to the cell body that we see of the neuron. This is where the nucleus is going to take place, or is going to be present, which is going to synthesize proteins. This is where membranes are going to be present and different chemicals are going to be present. You can see little mitochondria in there. You can see your ERs, your endoplasmic reticulums. And so the structures then of the cell body include the rough ER, which you can see here. In this picture, it's these little blue. So see right around the nucleus, you have those little lines, right? So that's your rough ER. The neuron cell body also has this small spherical nucleus with a nucleolus inside of it, which is the smaller little dot inside of there. The cell body itself contains some pigments, and in most cell bodies, the plasma membrane is part of the receptive region that receives input from other neurons. So anywhere basically outside of the cell body is going to be considered receptive regions. And so most of the time, the plasma membrane of the cell body itself is going to be part of these receptive regions. Now, the name of cell bodies, it depends on which nervous system we're talking about. Because in the central nervous system, when you have clusters of cell bodies together, they're called nuclei. In the peripheral nervous system, however, when you have these clusters of cell bodies, they're called ganglia, okay? So just some important terminology to point out to you before we move on. So as I previously mentioned, in addition to having a cell body, the neurons also have processes. And these are just arm-like projections, essentially, that are extending off of the cell body. So all of these branching tree, sort of limb-looking structures, and then also this projection down here. The central nervous system is going to have both the neuron cell bodies and their processes, but the peripheral nervous system mostly just consists of the processes, either dendrites or axons. And the names of these processes as bundles just depends on where they're located. So when you have bundles of neuron processes in the central nervous system, those are called tracts. So say, for example, you had a bundle of axons that are all rolling together, and I'm kind of moving my mouse over an axon here. If you had a whole bunch of these bundled together, this would be called a tract if it was present in the central nervous system, so if it was in the brain or the spinal cord. However, if it was in the peripheral nervous system, a bundle of axons, for example, together is called a nerve, All right, so we're all pretty familiar with the idea of a nerve. A nerve itself is just bundles of processes together in one peripheral nervous system structure. So again, we have two types of processes, dendrites and axons, and we're going to talk a little bit about each of these. So beginning with dendrites, dendrites are all of these limb-looking, branch-looking structures located directly off of the cell body in this particular case. Motor neurons can contain hundreds of these short tapering processes. And these processes, they have the same organelles as what are in the cell body. And then they also contain dendritic spines, which are basically these appendages with little bulbous spiky ends. So down here in this picture is where you can see a dendritic spine. So coming off of these dendrites, just draw a little piece in here for you. Coming off of each of these dendrites, you would have little spines, okay? The function of dendrites is to receive input, right? So this is the receptive region of the neuron, okay? So messages are going to enter. So messages are going to come in and they're going to land on these dendrites, okay? And then the dendrites are going to take those messages and send them to the cell body, okay? So dendrites receive the messages, the input region, and then they convey those messages towards the cell body as graded potentials. All right, and remember back to the muscle tissue lecture, a graded potential means that different amounts of stimuli are going to produce different responses. So if you have a whole bunch of signals coming in to the dendrites and then being transmitted to the cell body, then you're going to have a way stronger response than if only one or maybe two signals are coming in, which might not produce a major response. Additionally, there's many brain areas where more fine dendrites are very highly specialized to collect information. Okay, so these are the information collectors. All right. In addition to the dendrites, we also have the other process called the axon. Okay, and so the axon is all of this stuff here, okay, that I just outlined by red. Now, each neuron is going to have one axon that starts at this cone-shaped area called the axon hillock, which is what you see here, okay? So here's the axon hillock. In some neurons, the axons are super short or they might completely be absent, but in others, the axon can actually comprise almost the entire length of the cell. For example, some axons can be over 1 meter long, right? So it's like over 3 feet long. Additionally, really long axons are called nerve fibers. Occasionally, axons will have branches called axon collaterals, but that would be, for example, I'll just draw one in. Sometimes you'll see, we're going to just branch off this way. Okay, and that would be an axon collateral. All All right, it might have an occasional branch, but axons in general, they profusely branch at their terminal region at the end. Okay, and they can have as many as 10,000 of these terminal branches. And the distal endings here are called axon terminals. Okay, so each branch has a terminal. So for all intents and purposes, we have 1, 2, 3, 4, 5 branches. Each one of those 5 branches would have a terminal on the very end of the branch. Okay. So in terms of their function, axons are the conducting region of the neuron. So they're going to take the information that was sent from the dendrites to the cell body. They're going to take that information, and then that information is going to be sent along the axon. Okay, so it's the conducting region. It's going to generate these nerve impulses and transmit them along the axolemma to the axon terminal. And the axolemma is just this region along this way, sort of between the hillock and the terminal. All of this would be considered axolemma. The terminal region, then, each axon terminal is going to secrete neurotransmitters, which are going to be released into the extracellular space. So if you remember back to the muscle tissue section, I'm just going to do a quick little drawing here just to remind us. If you guys remember back to the motor end plates, and remember that our motor end plates, these neurons here, had these little synaptic vesicles which contained acetylcholine, that once there was a change in membrane potential, the acetylcholine would be released out here into this extracellular space, where it would then travel over to the muscle tissue, and it would land on a receptor. So here's our acetylcholine dots. Remember, acetylcholine would go and land on its receptor, which then would cause some kind of change to allow sodium and potassium to switch places and do all that. So that acetylcholine then is an example. Acetylcholine was an example of a neurotransmitter, which got released into this extracellular space, this gap between the neuron and the muscle cell. Okay. In addition, it's also good to understand that axons, they carry on many conversations with different neurons at the same time. And axons rely on their cell bodies to renew the proteins and the membranes that are present on them. If axons are cut or if they become damaged, they can very quickly decay. So from on that last slide, you saw all of these little bean-shaped, clear-looking things along the axon. What you were seeing is actually the myelin sheath. And so when looking at sorry about that. When looking at a neuron itself, I'm just going to draw one here really quickly. They're all the dendrites. And then you have the axon coming to the terminal, okay? So this is my 2-second drawing of a neuron here. What you can see wrapped around each of these segments are these myelin sheaths, okay? And myelin, essentially what myelin does for the neuron is that it protects and it electrically insulates the axon. And because of this, what can happen, this is a great thing, is that as we are receiving, transmitting an electrical signal, that electrical signal, instead of having to travel the entire way, it's actually just able to jump from one myelin sheath to the next. All right, and these are actually, this is actually the way that we can speed up nerve transmission. So kind of going along with this, and then this picture over here, I guess I should point that out to the right here, is just how myelin sheaths wrap around axons. So the yellow part here is the axon, and then this is the formation of the myelin sheath. It starts to do this coiling motion till it coils and coils and coils and becomes thick enough. Now, as far as myelin goes, it's not present on every single neuron. So we have myelinated fibers, which have the sheath around them, and this is usually present on really long or large diameter axons. And this is also what makes up the white matter of the central nervous system. However, we also have non-myelinated fibers too. And non-myelinated fibers, they don't have this sheath on them. And so therefore they conduct impulses more slowly. And an example of this would be the gray matter in the central nervous system. These are non-myelinated fibers. Okay, so now that we know a little bit about neurons, we need to talk about some classifications. First and foremost, neurons can be classified structurally in 3 different ways. They are either going to be multipolar, and multipolar neurons have 3 or more processes. So they have one axon, and in this case, this one has 1, 2, 3, 4, 5, 6, 7, 8, 9 dendrites. Okay, so multipolar just means that they have 3 or more processes. So one axon and then everything else dendrites. This is the most common and the major neuron type in the central nervous system. The second type here are bipolar neurons, and bipolar neurons have only two processes. Okay, and when I say 2 processes, I mean coming off of the cell body, you have to count the number of processes coming directly off of the cell body. And so what we see here is, here's our cell body. You have one process coming off this way and one process coming off this way. So this one's bipolar. It has one axon and one dendrite. These are very rare, but they are present in places such as the retina, so in the eye, and then also in the olfactory mucosa, which is the smell receptors. Okay. Finally, we have unipolar neurons. Unipolar, they're also called pseudo-unipolar, and they only have this one T-like process, okay? And these are also bizarre because they actually have two axons. So if you look at, again, remember we're measuring from the cell body. Here's our cell body. We have only one process coming off of the cell body. So that's where the uni and unipolar comes from. And then we have two axons. So we're going to have one axon splitting and going that way. And then we have another axon going this way. Okay. And these axons are going to have sort of different names here. We're going to have the peripheral or the distal process, and the peripheral process is going to be the one that's associated with the sensory receptors. And then the central process or the proximal process over here on the right is going to be the one that enters the central nervous system. Okay, so those are the structural classes of neurons, and structural just means how they look. So these are the three different ways that they might look, though I will say that carefully because there are always exceptions. Some of them might look slightly different, but these are the three general classes of the structurally classifying neurons. However, we also classify neurons, I'm going to just, here's a table sort of going through the three different structural classes of neurons, and you guys can take a look at that on your own. I just wanted to put that in there for you to sort of help organize everything in your mind. So in addition to, as I was saying before, classifying neurons structurally, we also can classify them functionally. Sorry about that. So we also classify them functionally. And functional classes are classes that are made up basically telling us how they work. Right? And so there's three types of functional classes, and these functional classes are based on the direction that the nerve impulse is going to travel relative to the central nervous system. And so those three classes, they're either going to be sensory neurons, motor neurons, or interneurons. All right, and you guys already hopefully have somewhat of an understanding about this. So sensory neurons are going to transmit impulses from sensory receptors towards the central nervous system. So for example, most unipolar neurons are sensory neurons that conduct their impulses along the afferent pathways to the central nervous system. Okay, so say, we're going to say we have sensory receptors here on our skin, all right? So we have some sensory receptors on our skin. And so whenever you feel something with your skin, so say you're feeling your clothes right now, that information actually gets sent to the central nervous system and tells the central nervous system that you're feeling something, okay? Essentially, all bipolar neurons are sensory neurons. And remember, these bipolar neurons are the ones that are pretty rare, the ones that are located like in the retina, for example. And the cell bodies of sensory neurons are located in ganglia, generally within the peripheral nervous system. Okay? So all of the cell bodies, for the most part, all the cell bodies then of these sensory neurons are located in ganglia that are in the peripheral nervous system. Okay. The second class are these motor neurons, and we already kind of talked about motor neurons, but let's talk about them a little bit more in terms of functional class here. So motor neurons take signals from the central nervous system and send those signals to effectors. Okay, so here's the spinal cord, a cross-section, sorry about that, of the cross-section of the spinal cord. And this red is the motor neuron taking information from the central nervous system, specifically here the spinal cord, and sending that signal out to an effector, in this case muscle. Motor neurons tend to be multipolar, and most cell bodies of motor neurons are located in the central nervous system, except there's some autonomic neurons that don't have cell bodies in the central nervous system. But for the most part, most of them have cell bodies in the central nervous system. Okay. And then this third and final functional class here of neurons are called interneurons. And inter usually means between. And so inter usually means between. And so these interneurons lie between motor and sensory neurons. And basically these are just little shuttle signals through these central nervous system pathways. And so if we have a signal coming in, okay, so let's do it like that. I'm gonna have this, have a signal coming in and synapsing here. This is gonna be our sensory input. It's gonna come in, it's gonna synapse on this interneuron, the one in green. The interneuron is then going to take that sensory information, turn it into a motor signal, and send the motor signal out via this motor neuron. Okay? So the interneurons then are the between neurons. And 99% of the body's neurons are actually interneurons, right? So these are little think of them as translators, okay? So they're translating this sensory signal that they're receiving into a motor signal, right? So our interneurons, these are our translators, okay? All right, so now we've already finished our conversation on neurons, and now we're going to talk about neuroglia, also called glial cells, and these particular cells function to support the neurons. The central nervous system has 4 types of neuroglia cells, including astrocytes, microglial cells, ependymal cells, and oligodendrocytes. And then the peripheral nervous system has 2 types of neuroglial cells, including satellite cells and Schwann cells. So let's talk about each of these individually. And we're going to start with astrocytes, which is one of the neuroglial cell types in the central nervous system. CNS is central nervous system. So astrocytes are the most abundant, versatile, and highly branched of all the glial cells. So they're shaped kind of like anemones. And what they do is they cling on to neurons. So this yellow thing back here is a neuron. They also cling on to the synaptic endings of neurons. And finally, they cling on to capillaries, which are just little tiny blood vessels. And the whole purpose of astrocytes, they have several roles, but they do things like they support the neurons and they brace them. They also play a role in exchanging nutrients between the capillary and the neuron. They guide the migration of young neurons to their proper places, and can control the chemical environment that's surrounding neurons. Astrocytes also respond to nerve impulses and to neurotransmitters, and research has it that we believe that they actually influence how the neuron is functioning and can participate in information processing in the brain. The second type here are the microglia. And these ones again are located in the central nervous system, and they are these small little ovoid cells with these little thorny processes coming off of them. And what these do, they touch and they monitor neurons. And they also tend to migrate towards injured neurons. So if you have an injured neuron, these microglial cells can actually evolve themselves and transform themselves to phagocytose any microorganisms or neuronal debris that's present. Okay, so these are basically like our little defense cells that are in the central nervous system that are sort of swarming around neurons, checking on them and making sure they're okay. The third neuroglial cell in the central nervous system that we're going to talk about are called ependymal cells. And these really range in shape from being squamous to columnar. And so I would call these ones in this particular picture being columnar. And they may or may not be ciliated, but the cilia, if they are ciliated, they actually beat, so they actually move, which helps to circulate cerebrospinal fluid. Okay, which is this fluid layer that is surrounding the brain and the spinal cord and protecting it. The ependymal cells line central cavities then of the brain and the spinal column, and they also function to form this barrier between cerebrospinal fluid and the tissue that the fluid is bathing in these central nervous system cells. Okay, so they're sort of forming a barrier between the CSF and the actual cells that are located within the brain and the spinal cord. Okay, these fourth types of cells that we're going to talk about are called oligodendrocytes. And I encourage you to say that word out loud a couple times because it's a very fun word to say. But these are branched cells whose job is to wrap nervous CNS fibers and produce and form those myelin sheaths. Okay, so the oligodendrocytes are the ones that are producing myelin sheaths in the central nervous system. Now we can move on to talking about the neuroglia of the peripheral nervous system. And the first cell type here we'll talk about are satellite cells. Satellite cells basically surround neurons in the peripheral nervous system, and they function similar to the astrocytes of the central nervous system. So if you can't remember what the astrocytes do, remember these are the ones that sort of latch on to the capillaries and the neurons, and they help with gas exchange and chemical environment. These satellite cells are doing very similar things, the only difference here being that they're located in the peripheral nervous system, okay, peripheral nervous system. The second and final cell type that we'll talk about here are Schwann cells. And Schwann cells, again, they're located in the peripheral nervous system. The Schwann cells are the ones that make the myelin sheath for the peripheral nervous system. So remember, in the central nervous system, the cells that made the myelin were called oligodendrocytes. In the peripheral nervous system, they're called Schwann cells. And these are absolutely vital to regenerating damaged peripheral nerve fibers. Okay, so they have the ability to regenerate damaged nerve fibers in the peripheral nervous system. Okay, these cells are very special in that way. So if you didn't understand this, I could have given you this example, but I didn't because I'm not the greatest with electricity concepts. But essentially what this is saying is that usually whenever, so say you have a short in your headphones, okay? And that short is, you can see the actual wires of your headphones, right? So you can see the little electrical wires that are belonging to your headphones. Sometimes what you can do to prevent yourself from getting electrocuted is you can wrap those electrical wires in electrical tape. Okay, and so this analogy is saying these wires are sort of like nerves, and if the wire is like nerves, then, and if these are peripheral nerves, then this electrical tape here that's wrapping the nerves is kind of like the Schwann cells that make the myelin to wrap up the nerves in the peripheral nervous system. Okay, so if you didn't understand that question and analogy, I just wanted to explain that. So that concludes the anatomy portion of the lecture, and I will stop this video here, and I will put up the video for physiology as a separate video. All right, so I will be back with the physiology video.